Thank you so much, David, for that really warm introduction. We are just so honored and humbled to be here, and particularly following Dr. McPherson. Congratulations to you on your award. It's certainly humbling, and we are here representing a very large team, which I'll tell you about in a minute. And we are really pleased to share with you some of our experiences in developing retinal gene therapy. We were fortunate to achieve many firsts, shown in this slide. The first reversal of blindness in a large animal model with retinal degeneration, shown here as Lancelot in 2000, after he had received a single injection of a gene therapy reagent in one eye. This is his third visit to Congress, where he spoke about the importance of biomedical research. And another first is the first child in the United States who received gene therapy, a retinal gene therapy, in 2009, 1 year after his injection, along with the team involved with that study. And then in 2017 was the first approved gene therapy reagent in the United States for inherited disease. This is LUXTURNA, and it was approved a year later in 2018 in the European Union as well. This has been an amazing experience for us. And before I go forward, I'd just like to mention that Al and I are co-authors on a patent for this work that was licensed to Spark Therapeutics, but we waived any potential financial gain long ago. And I'd also like to mention that we represent a large team of talented people. This is actually a partial list, and showing individuals involved in the various studies, including leading up to LUXTURNA administration centers, of which we have two, one at Children's Hospital of Philadelphia, CHOP, and one at UPenn. And it doesn't mention all of the people at Spark Therapeutics and the people who helped us present at the FDA Advisory Board and all the regulatory bodies, including the FDA, who were extraordinarily helpful along the way. Retinal gene therapy is alive and well. There are more than 1,100 different patients who have been enrolled in retinal gene therapy clinical trials. There is now, as I mentioned, one approved retinal gene therapy, that's LUXTURNA. LUXTURNA and other reagents have been administered at numerous centers around the United States and around the globe, shown in the map with these red spots where you can see these various centers. And now there are at least 13 different centers of excellence where this gene therapy, this approved gene therapy, is administered, shown by these black stars. Importantly, there are many more gene therapy clinical trials being planned, and we hope that LUXTURNA is just the first of many successful gene therapies. So where did it start? In 1985, when Al and I were glowing from the success of our first gene transplant procedure, the birth of our child Sarah in medical school, when we were in medical school, Al had asked me, had popped the question, do you think we could transfer genes to the retina? Well, he had been working in the lab of Elliot Burson, who ran the first lab to study human retinal degenerations. He knew that I had been involved in a collaboration with Dr. French Anderson, who had been planning what turned out to be the first approved human gene transfer study for an immunosufficiency. And in the glow of our success, I said yes, but I didn't tell him about all the problems. We had no genes to work with. There were no animals that had been described, no natural history, no reagents with which to carry out somatic gene transfer. In fact, nobody had tried that. No surgical methods, many no's. But thanks to pilot grants from groups that took a big risk on us, including the RP Foundation, which is now the Foundation Fighting Blindness, and the Associated Retinal Consultants in Royal Oak, Michigan, we took a start. The first step was to test non-viral agents ability to deliver genes to the retina. And you can see in this eye here with the blue cells that this actually did work, but this only worked for about 2 weeks. And so we were fortunate to then move to be able to move to different recombinant viral vectors to deliver DNA to the target cells in the retina. And the first steps, we were the first ones at University of Pennsylvania to actually test these vectors. Because there was little known about the safety of these vectors, we were forced to work in a biohazard level 3 facility, which involved gowning up and working in biosafety cabinets. Over the years, the safety information and the success of different viral vectors has become we know more about it. We know that adeno-associated virus, or AAV, works really well at delivering genes to cells in the retina. Shown here are cells turn green thanks to AAV delivery in the neural retina. And now people generally inject these viral vectors just with mask and gloves on, none of this other biohazard level 3 stuff. We also served as DNA delivery vehicles in the process, starting with moving equipment into the biohazard level facilities and continuing now moving microscopes from facility to facility and institution to institution. It keeps us healthy and strong and gives us some quality time together. So let me tell you a little bit about this disease. This is an autosomal recessive condition caused by 2 different mutations in the RP65 gene. RP65 encodes a protein standing for retinal pigment epithelium 65 kilodalton protein, which is involved in the retinoid cycle. And without a normal copy of this protein or gene, the cells in the retina cannot see. So in children with this condition, as well as animal models of this condition, there is night blindness is an early symptom. Even worse than that, there's progressive impairment as the cells degenerate in the retina. And you can see that there is often nystagmus both in the children and the dogs with this condition. We we carried out our first proof of concept study with this, with our first construct in the dog I told you about. This is Lancelot, and shown here, his electroretinograms went from basically flat, even at the highest light intensities, to robust, mirroring what is seen in a normal-sighted dog. And this persists, importantly, through the lifetime of this animal. We explored other outcome measures, including nystagmus testing. Shown here is the garment used for this testing, which looked like Fredericks out of Hollywood. And we also looked at outcomes such as pupillary light reflex, where in this animal who was illuminated in the uninjected eye, you see there's no pupillary light reflex, whereas when the light is switched to the injected eye, there is a brisk response. We tested the ability of these animals to see outside, and shown here is one animal who saw a squirrel and then escaped. Fortunately, we caught this dog and he did not lose him, learned our lesson from that. However, we realized early on that we could not use a reading test for these animals. There's one dog that successfully was able to read his own journal, but we thought the mobility test was more reliable. Shown here is the outcome for one dog pre-injection, and then after injection, this same dog is walking around without bumping into objects, is wagging his tail, is much more visual. And the results of these studies gave Al the idea, well, maybe we should plan an outcome measure test similar to this in humans. So I spent a month of August putting together a mobility test in our driveway, and this was one of our experimental outcome measures in phase 1 clinical trial. As you can see here, this worked quite nicely in, again, the first boy in the United States to be enrolled in a retinal gene therapy study. We also used this test in testing the safety and efficacy of repeat administration. This is to the contralateral eye. Shown here is Venus, one of the dogs who, along with her son, Mercury, participated in these studies. After they completed these studies, we adopted them, and Venus lived out her life to age 12, which is a long time for these large dogs, still seeing at the very end. So how did this work so well? There were a number of steps we took early on which helped make sure that this was going to be super efficient. One was to develop a supercharged reagent containing a transgene cassette which turns on robust levels of the RP65 gene, allows high levels of expression. We packaged it in a viral vector known as AAV2, and we made sure that every virus we delivered was functional, i.e. we got rid of the duds, the vectors which did not contain an RP65 gene cassette. We also did some studies with devices which showed us some surprising results that if we did not add a surfactant to the reagent, more than 75% of the reagent stuck to the inert surface of the syringe, whereas if we added surfactant, we actually delivered the total dose that we planned. We also spent time developing the surgical injection procedures. Rather, Al spent that time. Alan Ho gave a very nice description in his talk earlier of these procedures, and we shared these what we had learned with other investigators. Shown here is a video from Jason Commander. So now let me introduce my colleague.